In details, fixing histopathology samples. Mean. Fixed sample in 1 day. With 10% formalin. Or 75% alcohol. Or Carnoy's fluid. To prevent autolysis. For possible living state. To give suitable texture for staining. While processing histopathology samples. Apply by dehydration histopathology samples with series dilutions of ethanol alcohol. As follow. 70%, 80%, 95% and 100%, to pull water from sample. After that, apply cleaning with xylene, or chloroform, or benzene, as showed in next slide. Whereas, embedding histopathology samples. Apply by pour fresh melted wax at 60 degrees Celsius in sample. And allow wax to settle, cool, and solidify surrounding sample. But be attention please. Paraffin wax volume should be 25 to 30 times of sample volumes. To support sample on slide without injury. And to preserve sample relationship to each other. As mentioned in next slide. Meanwhile, sectioning histopathology samples. Apply by using knife and microtome machine to cut 4 to 6 micron thick section to mount in glass microscope slide, as mentioned in next slide. So, staining histopathology samples. Apply by 6 steps. 1. Remove paraffin from sample by dissolving with xylene. 2. Rehydrate section in descending series dilutions of ethanol. As follow. 100%, 96%, and 80% for 1 minute. 3. Apply stain for 10 minutes. 4. Remove excess dye by tap water. 5. Dehydrate section in ascending series dilutions of ethanol. As follow. 80%, 96%, and 100%, for 1 minute. 6. Clear slide with xylene and enjoy examining with microscope. As mentioned in next slides. Finally, this is section. Show anatomy of tissue as close as life structure, including cell cytoplasm, cell nuclei, and collagenous connective tissue. Thanks for attention. Please like and share and subscribe Genetic Teacher videos and channel. And for any question, please write comment.